This is the most interesting staining method, the Golgi staining, which is able to visualize not only the cell body or the processes, but together. The only disadvantage of this staining is that we are not able to predict in advance what cells will be visualized. So we have to double check all the preparations and hunt for cells. In this picture, you see a nice Purkinje cell in this region, upside down. The dendrite tree is visualized quite well, and this extends in the whole molecular layer, and the carrot-like cell body is also quite well seen. So the dendrite tree of the Purkinje cell determines the length, the extension of the molecular layer. Similarly, if you look at the upper part of the folium, the Bergman glia cells, the processes of which determine exactly the same thickness of the gray matter of the cerebellar cortex, so the molecular layer. This is what I suggest to see first, to be oriented where the thickness of the molecular layer is. With this magnification, you can see much better the previously mentioned Purkinje cell, the dendrite tree in the molecular layer, and the carrot-like quite large cell body. Under the Purkinje cells, in the remaining gray matter, you see quite messy cells. These are astrocytes, because this is still in the gray matter. These are protoplasmic astrocytes. More inside, where you see this part of this slide, this is white matter. So where you see again messy cells with not so clear contour, these are astrocytes, but in this case fibrous astrocytes. On the right side of this section, you see some Bergman glia cells extending to the whole molecular layer. The cell body corresponds to the Purkinje cell layer, and the processes are found in the molecular layer. In this part of the preparation, you see the Purkinje cell quite well again, and a little bit deeper, under the level of the Purkinje cell layer, we see a quite large perikaryon, but irregular perikaryon. This belongs to a Golgi cell. In the upper part of the slide, here, just above the perikaryon of the Bergman glia cell, so it is already in the molecular layer, you see a basket cell. And otherwise you see on the right side of this photo Bergman glia cells upside down, extending through the whole molecular layer, and otherwise you see again the quite messy cells, partly in the gray matter. Then these are protoplasmic astrocytes, partly in the white matter. These are fibrous astrocytes. In this part of the photo, we see a quite huge and irregular cell body with the processes. This is a Golgi cell. This is just outside the white matter, which is this. So it belongs to the gray matter, to the innermost layer, which is the granular layer. And across, so on the other side of the white matter, we see again in the gray matter, in the granular layer, tiny cells. These are the granule cells. In this part of the photo, we see a basket cell with its transverse running axon and extension, and we see underneath some little granule cells, and the thickness of the molecular layer is represented by a Bergman glia portion. In this part of the preparation, in the upper part of the molecular layer, we see a stellate cell with its cell body and processes. Here you see the extension of the Bergman glia, showing that these cells, the stellate cell is located in the upper half of the molecular layer. In the deeper gray matter layer, in the granular layer, we see some granular cells, and the other messy cells in the gray matter are the protoplasmic astrocytes. In this part of the photo, we see two beautiful basket cells. One is especially very nice because of the extension of the dendrites and axons. The other one shows also transverse running axon, and they are located just above the cell bodies of the Bergman glia cells, so they belong still to the molecular layer this way, located in the deeper part of the molecular layer. Underneath, in the granular layer, we see a nice granule cell with 4 or 5 bird claw-like processes. The other cells are mostly astrocytes. Those which are in the gray matter, the protoplasmic astrocytes. Those which are in the white matter, such as these, are the fibrous astrocytes. In the middle, at the bottom of the picture, we see a nice, maybe stellate cell, because this is located in the upper part of the molecular layer.